Hello, students. In this lecture, I'm going to be talking a little bit about the nervous system. So I'm going to be kind of introducing both of the major divisions of the nervous system, the central and then the peripheral nervous systems. And then I'm going to be talking about neuron physiology and how action potentials or nerve impulses are sent. Okay, so let's look at the functions of the nervous system. Remember that the nervous system is really specialized for communication. And we see that in these functions, of course. So sensory input, that is just gathering information. Basically, this relies on the presence of sensory receptors. Now, these can be receptors that monitor changes in temperature, pressure, pain, photo in the case of the light receptors in our eyes, the cone and the rod cells. So there's a lot of different types of sensory receptors, and they're all specialized for a particular purpose. So these receptors are what monitor changes. Those are called stimuli that are occurring both inside and outside the body. So for example, a type of stimulus that might occur inside the body would be pain from, say, lack of blood flow to an organ. A stimulus that's occurring outside the body, that could be anything from a smell to something that you're seeing to a sound. So a stimulus can just be anything that our body can detect. And again, that's going to be the sensory input function. Number 2 is going to be integration. So once we are able to sense this stimulus, what are we going to do with that information? That's where integration comes in. So the nervous system is really going to process and interpret sensory input and then decide whether or not an action is needed. And we can also basically picture this as a neural circuit. So remember that this is a poor representation of a neuron, but remember that you have these stellate-shaped cells. The soma or cell body is up here. Then you have the axon. The message is in the form of a nervous impulse or an action potential. So that's going to be sent along the length of the axon. And so this would be your sensory input neuron. That neuron is going to be the sensory receptor. It's going to synapse with what's known as an interneuron. So an interneuron is going to be found within the central nervous system, either the brain or the spinal cord. And it's going to be in synaptic contact with the sensory neuron. So the interneuron is in turn going to be in synaptic contact with the motor neuron. So the motor neuron is going to extend all the way to whatever effector organ is going to be making a change in the body. So let's talk about something that we've already discussed for this class, which would be skeletal muscle cells. So remember that your skeletal muscle cells form synapses with your motor neurons. So this is essentially how we can consciously, physically respond to certain types of stimuli. So this would be your sensory neuron, this would be your interneuron, this would be your motor neuron, and then whatever tissue or organ is in synaptic contact with your motor neuron is going to be the effector organ. So that's going to be muscles or glands. So motor output is the response or effect. It activates muscles or glands. And these can be skeletal muscles, they could be cardiac muscle, smooth muscle, glands. What type of tissue it is going to reflect whether that is an autonomic or automatic response to a stimulus or a conscious response, and that would be skeletal muscle. So those are the main functions of the nervous system. Okay, so basically, here's an example. We have sensory receptors The sensory receptors in our eyes are called photoreceptors, and they are specialized to send nervous impulses when light enters the pupil. So we have light is going to cause a reaction with our photoreceptor cells, the rods and the cones, and nervous impulse is going to be sent. That's a sensory input. We have integration, so processing that's going to take place within the central nervous system, within the brain, and then motor output. So in the case of this example, this is a skeletal muscle, of course. This is under voluntary control. So if we're responding to something that we're seeing, making a conscious decision about it, and going away from or towards whatever we're seeing via our skeletal muscles, that would be an example. And of course, this process is occurring in many different parts of our body with many different sensory receptors. It doesn't have to be a conscious process. It can be like a reflex arc, for example. If you've ever gone to the doctor's office and they've tested your patellar reflex by tapping basically just below the patella, if you have a functioning patellar reflex, your leg is going to involuntarily kick up in response to that. But the same basic pattern would exist. We have sensory input, basically sensing the pressure from the tap. Integration would be an interneuron within the CNS, and then motor output to the muscle. All right, so let's look at the two main divisions and then the sensory and motor divisions within those. We have our central nervous system, which is the brain and spinal cord, and then our peripheral nervous system, which are the cranial and spinal nerves. So essentially, as soon as these nerves, these bundles of axons, exit the brain or spinal cord, they're considered part of the peripheral nervous system. And those nerves are how we send communication from the brain and how information goes from parts of our body to the brain. So we have the motor division here. Again, motor, think output. And another term for this is efferent. So motor output, we have our autonomic division of the nervous system. This is involuntary. So this is going to be controlling cardiac and smooth muscle cells and glands. And then we have the sympathetic and the parasympathetic divisions. Sympathetic is known as fight or flight division, parasympathetic rest or digest. And what this essentially means is that most of the structures in our body, most of the organs within our body are dually innervated. So dual innervation refers to both sympathetic and parasympathetic nerve fibers coming into contact with the same organs. So, for example, we have our heart. If the primary control is coming from the sympathetic division, that's going to increase the heart rate. And similarly, we can also have parasympathetic control over the heart, which would decrease the heart rate. So that is dual innervation. Most of our organs are dually innervated. So these two systems often oppose one another in terms of their action, but sometimes they work together. Okay, so that's the autonomic or involuntary division. And then the somatic division is voluntary, and that's going to be our skeletal muscles. So that is all having to do with motor output. The sensory or afferent division is just how we gather information. So those are going to be our sense organs. Those are going to be things like thermoreceptors. Those would be nerves that sense changes in temperature. Proprioceptors, which basically keep track of body position in space and help with basically keeping track of body and limb movements. They could be nociceptors, which are pain receptors. They could be photoreceptors, like I've already talked about. Basically, sense organs, any sort of specialized or free nerve ending that is capable of sensing a stimulus and responding appropriately. Let's look at the structural classification in a little bit more detail here. We have our central nervous system, generally just abbreviated CNS. The organs involved in the CNS are going to be your brain and your spinal cord, and the function is going to be for integration. It's the command center, essentially. It's going to be interpreting incoming sensory information, and then it's going to be issuing outgoing instructions. So again, these are the interneurons that are involved in the CNS. And then the peripheral nervous system, or PNS, basically this is comprised of nerves extending from the brain and spinal cord. And you might also see an associated term, ganglia, or ganglion, just singular. These are clusters of cell bodies. So remember that your cell body, those are your stellate cells. You have your long axon that sends out your action potentials, comes into synaptic contact with other tissues. If you have a cluster of these, that's going to be a ganglion. Whereas if you have many of the axons bundled together, that's going to be a nerve. So it's not just a single axon, it's going to be many axons that make up a nerve. Similar to with a ganglion, you're going to have many cell bodies concentrated in one location. And you'll see these ganglia basically right outside the vertebral canal. So right outside the spinal cord, you'll see swellings that are concentrations of these cell bodies. And then the nerves that are extending out from that are going to be bundles of axons. So the spinal nerves are going to carry impulses to and from the spinal cord, and then cranial nerves are going to carry impulses to and from the brain. Let's see, and functions, of course, going to serve as communication lines among sensory organs, the brain and spinal cord, and glands or muscles. So the sensory and motor division, basically these are ways to classify the nervous system from a functional perspective rather than just structural. So the sensory or afferent division are going to be your nerve fibers that are carrying information to the CNS. The somatic sensory fibers carry information from the skin, skeletal muscles, and joints, and then the visceral sensory fibers are going to carry information from visceral organs. And a little bit of terminology info here, fibers, that's basically referring to axons. So they have this long fibrous appearance, so they're oftentimes referred to as fibers, or nerves. Sometimes that's used interchangeably with nerves too. And then the motor or efferent division is going to be your nerve fibers that carry impulses away from the CNS to the effector organs. So sensory is on the input side, motor is on the output side, and of course in between we have our interneurons within the CNS. If we're looking at the motor division in a little bit more detail, we can break that down into two subdivisions. Somatic nervous system, which is voluntary. This is going to consciously control skeletal muscles. And then the autonomic nervous system, which I mentioned previously with that little kind of map of the nervous system. This is going to automatically control smooth and cardiac muscle as well as glands. And it's further divided into the sympathetic and parasympathetic nervous systems. Okay, so like I said at the beginning of this lecture, nervous tissue is made up of two principal cell types. We have our supporting cells, which are the glial cells. Now, this particular author describes them as resembling neurons. I haven't really found that to be the case. From looking at our histology slides, the glial cells are generally a lot smaller, and they're a lot more numerous as well. So there are also many different types of glial cells, so it really kind of varies depending on which type of glial cell you're referring to. Glial cells are not able to conduct nerve impulses, so these are not the cells that are actually communicating. They do, however, serve various functions in support of neurons. So they form some structural frameworks, they form part of the blood-brain barrier, for example. They help in terms of immune response within nervous tissue. They produce cerebrospinal fluid in some cases. So they're not able to conduct nerve impulses, but they are going to serve a number of different purposes that are essential for this tissue. And glial cells are never going to lose their ability to divide. So most of the cell types in our body are able to undergo mitosis. There's a few exceptions to that. Red blood cells, of course, are amitotic. They're produced in the red bone marrow, not through mitosis. Glial cells are not going to lose their ability to divide, unlike neurons, which are the ones that are actually specialized for communication. So, kind of one of the implications of this is with brain tumors. If neurons are not able to mitotically divide, how do people develop brain tumors? That's because the glial cells are giving rise to tumors. All right, so let's just briefly look at the glial cell types. We have astrocytes. These are going to be the most abundant and versatile of the glial cells. We have microglial cells, which are phagocytes. That means they are going to be dispersed throughout nervous tissue, and they're going to consume waste products and potential pathogens. We have ependymal cells, which line cerebrospinal fluid-filled cavities, such as the ventricles of the brain. So they're actually going to be producing some of that cerebrospinal fluid. We have oligodendrocytes, which are found in the CNS. These are going to form the myelin sheaths around nerve fibers within the CNS. Primary purpose of that is going to be to increase conduction velocity of the nervous impulse. And remember that white matter within the brain and spinal cord is going to be myelinated. That means it's going to have these oligodendrocytes on the axons, and that helps to insulate the fibers, helps to conduct action potentials more swiftly. And then in the peripheral nervous system, we have a different type of glial cell that forms myelin sheaths around axons. So functionally, they're going to be pretty similar. Structurally, they're going to be a bit different. But satellite cells and Schwann cells are going to surround neurons within the peripheral nervous system. Oh, and I forgot to mention with astrocytes, one of their main functions is to form that blood-brain barrier. So they have extensions called perivascular feet. Which is essentially what you're seeing here. They are going to wrap around capillaries. So capillaries are the exchange vessels for the cardiovascular system. So this is how nutrients and gases are delivered to tissues, and then other gases and waste products are picked up by capillaries. The perivascular feet help to seal off intracellular clefts between the endothelium cells of the capillaries, and that helps to provide a line of defense between the blood supply and the brain. So it's it's basically a protective function for the brain, and that again is part of the blood-brain barrier. Okay, so the neurons themselves, another term for these, of course, are just nerve cells. These are the cells that are specialized to transmit messages or nerve impulses. The major regions of all neurons, we have the cell body, of course. This is where the nucleus is, and it's the metabolic center of the cell, so most of the organelles of the cell are going to be found within the cell body. And then we have the processes that are fibers that extend from the cell body. And those are going to be known as dendrites. And then, of course, the axon. So let's take a look at a nice illustration here of a single neuron cell. So the cell body, basically this entire region is your cell body. These dendrites are the tree trunk, not really tree trunk, really more like tree branches that are coming off of the cell body. The axon hillock, this is actually where a nerve impulse originates. So there are a lot of sodium gates here. Okay, so as I was saying, the axon hillock is where there are a lot of sodium gates. So remember that sodium is Na+, and this is actually really important for actually conducting a nervous impulse. Because in order to send a nervous impulse, we have to depolarize the cell membrane. And this region of the neuron has enough sodium gates that you can flood the inside of the cell with positively charged ions and depolarize the cell, meaning there's going to be less of a difference in charge between the inside and the outside of the cell. And I'll talk more about that later. But again, that's going to happen at the axon hillock. And then once that nervous impulse starts at the axon hillock, it's going to be sent down the length of the axon. So what we're seeing with this blue material, this is going to be your myelin sheath. And remember, there's two different types of glial cells that form the myelin sheath: oligodendrocytes within the central nervous system, and then the Schwann cell within the peripheral nervous system. So this would be representing a neuron within, say, a cranial or a spinal nerve, not within the brain or spinal cord. And we're not going to get into too much detail with the structure and properties of conduction, but there's the gap basically between the cell, or between the portions of the myelin. This is going to be the node of Ranvier. And then, of course, the axon terminal, this is where neurotransmitter is stored. These are chemical messengers. And once the nerve impulse reaches the axon terminal, neurotransmitter is going to be released. It's going to diffuse across what's known as the synaptic cleft, and it's going to be received by specialized protein receptors within the cell that this neuron is synapsing with. So neurotransmitters essentially are going to transmit the nervous impulse to the next cell. And here's an actual image of a neuron. Basically, looking, I accidentally skipped over that slide, but looking at the actual images of the neurons, it's never as nice and neat and organized looking as the illustrations in a textbook. It's going to be difficult to tell the dendrites from the axons, for example. Okay, so let's talk a little bit more about the processes of neurons. The dendrites are going to conduct impulses toward the cell body. A neuron may have hundreds of dendrites, and these dendrites, of course, can be in contact with other neurons, other cell types, etc. We have our axons that are conducting impulses away from the cell body. So this is really where you see an action potential itself. So neurons have only one axon arising from the cell body at the axon hillock. The axon is going to end in axon terminals, which contain those vesicles with neurotransmitters. And then the axon terminals are going to be separated from the next neuron by a gap. And again, that is known as the synaptic cleft. So it's just a small space that separates the axon terminal of one neuron from the next cell in this network, basically. Okay, so I got ahead of myself there with the synaptic cleft. That's the gap between axon terminals and the next neuron. Keep in mind, though, that if we're not talking about a network of neurons, we can also have our motor neurons in direct contact with, say, muscle fibers. That would be the neuromuscular junction. So the synaptic cleft is not only found between neurons, it can be found between a neuron and whatever cell type you're coming into contact with. So the synapse is just the functional junction between nerves where a nerve impulse is going to be transmitted. All right, so myelin. Again, myelin is going to be formed by either oligodendrocytes or Schwann cells. It's a white fatty material that covers axons. So it's mostly lipids. It's going to protect and insulate fibers. And the main function of the myelin sheath is going to speed nerve impulse transmission. So in the case of the Schwann cell, you basically have this pattern of portions of this Schwann cell that are wrapping around the axon of the neuron. And that's going to form that myelin sheath. Okay, so these are some terms that most of these you've heard of before, but let's just review some of the additional terminology associated with neurons. Nuclei. So this can be kind of tricky because, of course, we all know that a nucleus is going to be kind of like the command center of a single cell where the DNA is. But you can also have centers of cell bodies that are referred to as nuclei. And in that case, it wouldn't just be the DNA, it would be the entire cell body. So, for example, there are a number of groups of cells within the brain that have specialized functions. Those would be considered nuclei. So these are just clusters of cell bodies within the central nervous system. Then we have the ganglia, which are collections of cell bodies that are outside the central nervous system. Again, basically you're going to see these little swellings within nerves, and those are technically ganglia. Tracts are going to be bundles of nerve fibers within the central nervous system. Great example of that would be the corpus callosum in the brain. So that's a tract of white matter. Nerves are bundles of nerve fibers within the peripheral nervous system. So they're essentially just the counterpart of tracts, but within the PNS. And then white matter versus gray matter. This is having to do with whether or not you're talking about the fibers or whether you're talking about mostly cell bodies and unmyelinated fibers. So the white matter generally is going to be white because of the presence of myelin, and gray matter is mostly unmyelinated, so it's going to have more of a dull brownish gray color. All right, so again, I've covered most of this already. but just to provide some more formal definitions, the functional classification of neurons, we have our sensory neurons, our motor neurons, and then our interneurons. So our sensory or afferent neurons are going to be carrying impulses from sensory receptors. Again, those are things like thermoreceptors, photoreceptors, nociceptors, any of the neurons that are specialized for actually responding to stimuli. It's going to carry that information via a nerve impulse to the central nervous system. Let's see, we have our motor neurons or efferent neurons, which are going to carry impulses from the CNS to viscera and or muscles and glands. And then we have our interneurons or association neurons. These are the cell bodies that are located within the central nervous system. They're going to connect the sensory and motor neurons. So really, if you're looking at it from a functional standpoint, interneurons should be the one listed in the middle because they are between sensory and motor neurons. Okay, so this is essentially showing a cross-section of the spinal cord. And it's showing how the spinal nerve is integrated within the spinal cord itself. So afferent transmission, meaning sensory transmission, we would have a nerve impulse coming in through this route. This slight swelling here would be a ganglion. So again, you wouldn't just have one cell body. This is just illustrating one cell body for the purposes of showing you how this is set up from a structural standpoint, but there would be many cell bodies. So again, we have nerve impulse being sent along the axon here, and that sensory information is going to enter the spinal cord at what's known as the dorsal horn or posterior horn. So see how you have kind of this butterfly-shaped or H-shaped area of gray matter here? This portion is known as the dorsal horn. So sensory information is always going to come in the backside, and you're going to have your interneuron within the central nervous system. So this is going to be your interneuron here, and then this is going to be your motor neuron. You have a synaptic contact here, so message is going to be transmitted to this motor neuron, and then nerve impulse is going to be sent out, what's known as the ventral horn or anterior horn. So sensory information, posterior, motor output, anterior, and you're going to have your motor neuron synapsing with the effector organs, muscles, and glands. Now think back to when we were studying histology. We were looking at some of the fundamental properties and characteristics of the different tissue types. Remember that functional properties of neurons include both irritability and conductivity. Irritability, not grumpiness. Irritability is just the ability to respond to a stimulus and convert it to a nerve impulse. Conductivity is going to be the ability to transmit the impulse to other neurons, muscles, or glands. So just how certain metals are highly conducive to the flow of electrons, our neurons are going to be highly conductive to nerve impulses or action potentials.